Okay, so we are looking at transport in plants, and basically we are looking at blood. Our major discussion here is going to be about blood. What is blood? What makes up blood? The functions of blood, and then we also discuss about the immune response of blood, components, and all that stuff. So, what is blood? We say blood is a pale yellow fluid consisting of cells suspended in a fluid known as plasma, and this blood is made up of two major tissues. One, the one we can see is called plasma. This takes the biggest percentage, and also 45%, which we call the corpuscles, which are blood cells. So basically, blood is made up of two substances, major substances. It's a pale yellow fluid called plasma, in which are suspended white blood cells, platelets, and red blood cells. So we are going to see this in detail. We'll see that if you happen to get your blood and centrifuge it in a machine called a centrifuge, we shall get the different components of blood. We shall have the bigger component, which we see down here, and this is where we have the blood cells. The biggest percentage of blood cells are red blood cells. Then the next is white blood cells, and then the yellow part, which is the fluidy part here of this bigger part, it's plasma. So when you get a blood smear and you put it onto a slide and you drop this blood into this, you realize that blood is made up of different components. As you can see, we have the red blood cells, which are the red in color and responsible for the color. Then we have the white blood cells with one big nucleus. We have one white blood cells with lobed nuclei. These ones are different nuclei, as you will see later. Then you see another white blood cell with a big nucleus, but this one is one. This is lobed. This one are many. These ones are many nuclei. So you see that blood is basically of two. It's basically of two parts: the plasma and then the cells. So the plasma is a pale yellow fluid. It's a pale yellow fluid in which blood cells are suspended. So plasma takes the biggest percentage, which is 55%, and then the cell content takes the 45%. So plasma has a lot of components, as we are yet to see, but when you check if blood is centrifuged, the cells precipitate, leaving the plasma as a spermatant. Here is a photograph of the blood smear taken down a micrograph. So when you pour this, so you see that most of the times it's plasma that appears in many cases, and then cells can come out. A few of them can be seen. So the big ones are red blood cells, which we can see here. The ones with the biconcave structures, and then these big ones with the nuclei that are visible are white blood cells, and they have three types. There are those ones whose nuclei is divided into three. We have the ones whose nucleus is lobed, is entire, and then those whose nuclei is lobed. The lobed nuclei is like divided into portions, but still attached. So this is what we see when we get a blood smear and look at the blood components. So we've seen the red blood cells, we've seen the white blood cells, we've seen the plasma. Then plasma transports carbon dioxide. If I may say, a plasma transports carbon dioxide from the organs to the lungs. Then plus other substances. For example, urea to the kidneys, other substances like digested food materials are all transported by plasma. So microscopic drawing of the blood smear here, if I can draw it for you, and I want you to label this component, and label these components here, which are we are looking at as small fragments of white blood cells. So as we proceed to see, you realize that the ones with biconcave structure, this is the red blood cell. The ones with this structure here are white blood cells, and also the ones which are fragments here are platelets. So the three types of cells make up the 45% of the components of blood, and then the 55% makes up the smaller components of blood, including proteins, amino acids, glucose, mineral ions, urea, and all other substances which make up plasma, and it's the transporting media. So as we proceed, we will see that soluble products of digestion from the small intestines to other organs are transported by plasma. So that's one of the functions, transporting function of plasma. Carbon dioxide from the organs to the lungs are soluble products of digestion from the small intestines to the organs, that is plasma. Urea from the liver to the kidneys, that is plasma. We also have plasma contains a variety of dissolved solutes such as urea, glucose, and hormones plus heat. It also contains plasma proteins that remain in blood as cells for all the time. So this is when we see the importances of part of the functions of blood. So contains plasma proteins that remain in blood all the time. When we go to when we go to so if you continue with white blood cells, we shall realize that white blood cells are made up of different components, which components involve the bigger ones whose nuclei are divided into lobes, which we call the lobular nucleus, and the entire nucleus. So the entire nucleus of white blood cells will involve, they have nucleus, that's one thing. So if we can delay, we can make the difference between white blood cells and red blood cells. White blood cells have nuclei, and they have different shapes as compared to red blood cells. Red blood cells lack a nuclei, and they are said to be. So red blood cells have different structures which lack nuclei when mature. They are many, that means they are numerous, and that increases the surface area for transportation of oxygen. However, white blood cells form part of the body's defense system against microbes, as we saw in our previous videos regarding transport, regarding defense, specific and unspecific defense of the immune system. So when you look at red blood cells again, in other words, we will see that they have no nucleus. They are packed with red pigments called hemoglobin. So red blood cells have pigments called hemoglobin in them. And this hemoglobin is very important. It has a high affinity for oxygen. So we say in the lungs, oxygen combines with hemoglobin to form oxyhemoglobin. And that's a form in which oxygen is transported. Oxyhemoglobin is the form in which oxygen is transported to the rest of the body tissues, where it is needed to work as a raw material for aerobic respiration, and carbon dioxide is dropped in there. So when we see in other organs, oxyhemoglobin splits into hemoglobin and oxygen. So oxygen diffuses into the body tissues. They move across tissues into the mitochondrion, where aerobic respiration takes place by simple diffusion, and then it keeps on to simple diffusion to join the mitochondrion, where aerobic respiration takes place. So we have design features of red blood cells for oxygen carriage. How is oxygen adapted to carrying, how are red blood cells adapted to carrying oxygen? Number one, they lack a nucleus when mature, making more room for hemoglobin. Hemoglobin is a conjugated protein that is made up of a protein known as globin with iron 2 called heme. So hemoglobin is a protein in conjunction with joined together with a nanoprotein known as iron. So the two increase the capacity of the red blood cells to carry oxygen from the lungs to the rest of the body tissues where it is needed. So oxygen is important in that process, and it's very beautiful and useful when it comes to carrying out the process of transportation of oxygen. So one of the adaptations that when red blood cells are mature, they lack oxygen. They lack a nucleus to provide room for space for hemoglobin. Then they have the donut shaped. They are donut shaped, which is a biconcave shape, giving a greater surface area for gas exchange. So they have a donut shape, giving a great surface area for gas exchange. And also, if you check them, so they are very many. The last ones, they are numerous to increase on the surface area for absorption of the oxygen gas. They are numerous to increase the surface area for transportation of oxygen gas. So lacking the nucleus, having hemoglobin, and then by concave shape and being many are the four adaptations that help the red blood cells to be in position to perform their function. So if we ask you for a question to state the adaptation of red blood cells to their function. Number one, we are referring to the structure of red blood cells related to the nucleus. To its function, sorry. The structure of red blood cells related to the function. One, they have no nucleus when mature to provide space for hemoglobin. They have a pigment known as hemoglobin that has a high affinity for oxygen. Number three, they are numerous. They are very many to increase on the surface area for absorption of the gas. Number four, they are biconcave in structure, or they have a donut structure that increases the surface area for diffusion of the gas. When we look at other fragments which are found in blood, for example, these ones here, which are not easily visible, those ones mark what we call platelets. Platelets are blood fragments that are responsible for blood clotting. Blood clotting is the first step towards healing of the wound, and therefore they are very much important. They are not colored, so their biological name actually is they are called thrombocytes. Thrombocytes. So thrombocytes is the biological name for platelets. The biological name for white blood cells, because they are not colored, so they are referred to as leukocytes. Cyte stands for cell. Leuco, colorless cell. So white blood cells are called white because they are colorless. Then the red blood cells, which are the majority and many on the smear, as we've seen, they are referred to as erythrocytes. Erythrocytes. It means that erythro means colored cytes cells, colored cells. So those are colored cells. So platelets have no nucleus as red blood cells are, and platelets help blood to clot at the site of a wound. So remember, blood clotting is the first step towards healing of the wound. So when you get to damage a tissue, expose a tissue to air, and blood gets exposed, what you call the response will happen, whereby the platelets together with vitamin K and calcium ions will produce an enzyme known as thrombokinase, which thrombokinase will be important in converting a soluble a soluble protein known as thrombin, prothrombin, which is supposed to be converted to thrombin. Then after converting prothrombin to thrombin, thrombin now will facilitate the conversion of soluble fibrin fibers in blood into fibrin, which forms a network of fibers that prevents blood from oozing out. So this is a simple discussion about platelets. So what do? They are important in the blood clots. So when we draw for you a table and we give you the specific number of platelets, then in the other side, in the other instance, it's more than the normal, you'll know that now there is a wound, if there is a wound whereby the platelets are produced to come and reduce on blood clotting, blood oozing out and clots blood. So when you look at white blood cells in immunity, because this video links up with the previous videos we've been seeing, so it's one way of helping you. When we come to white blood cells, if pathogens gain entry, because we say they are responsible for producing or helping in immunity, that is defending the body against against diseases. So when a pathogen gains entry into the body, two types of white blood cells attack them. We have the one which we see here, and then the one which we see. So the one with one big nucleus, which is large, they are called lymphocytes. And these lymphocytes are produced in the lymphatic system, and they are responsible, they act as stem cells. They work as stem cells. Then the ones which have lobed nucleus are referred to as the phagocytes here. The phagocytes here, they are responsible for phagocytosis, that is, they attack the pathogen, engulf it, and feed on it. Going back to the lymphocytes here, lymphocytes are like stem cells, so they are undifferentiated cells which can differentiate into two types of cells. The one type of cell from lymphocytes will grow from bone marrows, and the other type of cells in the lymphocytes, when they differentiate, will grow from the thymus gland. So the other type of cells which grow from bone marrows will be responsible for producing antibodies. Antibodies, these are structures or the body defenders that attack the pathogen and help to disable and destroy it or represent it to the destroyers, which are the phagocytes for feeding. So the B lymphocytes are the ones which grow into the bone marrows, part of the lymphocytes. Then the other part which grows from the thymus gland, because thymus is T, so they are called T lymphocytes. And T lymphocytes are responsible for producing the CD4 cells, which are responsible for capturing and presenting pathogens for phagocytes to feed on. So this is how amazing our body system is. So these antibodies also, the B lymphocytes produce what we call stem cells. Stem cells will give us the long immunity, which we call the active immunity, whereby if you suffer from a disease for the first time, it will treat you bad, but the lymphocytes will copy it, produce specific antibodies, and also produce the long memory cells, which will be used for reference during any time of second infection, as we're here to see. So they also produce what we call antitoxins. Antitoxins, these are substances, these lymphocytes produce a substance that counterattacks the toxin a pathogen may produce. For example, we have some pathogens that produce toxins which result into allergens. You get allergies, you get responses which are not good all over the body. So what happens with this man of ours? They will help us to find. They will produce an antitoxin, as you hear the word antidote, that will counteract the effects of the effects of this toxin, and then neutralize you, heal before you even get sick. So phagocytes, they feed by phagocytosis. These are white blood cells that we can find in blood all the time, and they engulf the pathogens and digest it as food. So the method of feeding is called the phagocytosis. This type of cells during phagocytosis are white blood cells known as phagocytes. They have a nucleus which is lobed. This is what we call a lobed nucleus. So it has one lobe, second lobe, third lobe, and fourth lobe. So that's why it's called the phagocytes. Now, when we look at the white blood cells, we have very many. As I've mentioned, that phagocytosis, you see this white blood cell, it will come and find these pathogens, which may be like bacteria, then it will come and engulf it. So this is how it has done it. So we got this. Now I get this white blood cell. Now encounters this and then engulfs and digests the pathogen by phagocytosis. So what has happened is pathogen engulfed by white blood cells, so it has fed on it. So white blood cells feed on these pathogens in one of the best ways of protecting the body against them. So when I come to my friend, Mr. Lymphocytes, I said these are stem cells whose role is they produce uh, they produce two types of cells. They differentiate into two types of cells. One type of cells of lymphocytes will grow from the thymus gland around the chest here, and then the other one will grow from the bone marrow. So the first lymphocyte that grows in the bone marrows, they are referred to as B lymphocytes. So B lymphocytes, their role is to produce antibodies. Antibodies, they fight against specific antigens that have a particular structure. So antibodies are specific in the defense. So they will help our body to defend itself on specific pathogens. It doesn't attack anything. For phagocytes, they can feed on any. But the plasma cells, which you call the antibodies, they are very specific in structure, as you can see. It produces them. So these antibodies produces the attack the pathogens or their toxins. So as you can see, this is one antibody, another antibody. They have a structure which we call the epitope. This structure here is complementary to the structure of the pathogen. That's where specificity comes in. As you can see with white blood cells here, which we call the phagocytes, they are not so much into how this looks. No. It just finds it, attacks it, engulfs it, and feeds on it. But for this one, the reason why we suffer from an infection and it treats us so ill for the first time, because our bodies have not produced specific antibodies fighting against that particular disease. The first time I will get attacked by this infection, my lymphocytes will take time to study, then produce the particular antibodies that are specific, and also to produce memory cells. Memory cells will provide me with what we call the long-term immunity, whereby if the same mistake happens and this same pathogen comes back in my body, this body will already have a photocopy, which we call the memory cells, from which we shall copy and produce more respondents and make our body strong and heal before we even get any. So antibodies have structures as we have seen them. They are this. So the other part of the thymus, which you call the T lymphocytes, they produce the CD4 cells, which are responsible for patrolling and also producing picking pro- okay, producing cells which are patrolling cells, okay, which can present antibodies or antigens to phagocytes for feeding. Now, what happens when I get the infection? Once they have produced antibodies against a particular bacterium or virus, with memory cells can produce them quicker and greater quantity if the pathogen returns, giving the body personal immunity against diseases. Look at this. I have this. The very first time I get the immune response, I get the infection for the first time, my body, the level of antibody production will be very low, as you can see. The level of antibody production is very low from here. Why? The pathogen is eliminated, but the damaging symptoms of disease will have already occurred. So all here, as you can see, the quantity my body was able to produce is low, and you can see the time taken from the initial infection up to when I healed from this disease. All that time, because this pathogen is appearing in your blood for the first time on your body, the body is producing, is trying to copy the structure of the pathogen, is trying to go to its memory, to its system cells, being lymphocytes, and tell them that, you know what, this kind of infection has come, it is so serious, we need to make antibodies that are specific to this. So with that time, you realize that you get all the clinical symptoms. You'll be sick. You'll be very sick and ill, for your information. If not, you may even die. But in that process, the body is not sleeping. It is trying to copy the structure and then also produce antibodies, plus the memory cells that will always stay after when the second infection comes. So, what happens if the second infection comes? We are here. The second infection now will come like this. So with the second infection, you can see I will have my immune response. You see the level of production is so big. So that shows that the same pathogen happened to attack this very individual the second time. The second time, it will find the immunity so steady. So as it infects like this, because of the previous damages, the body was able to produce memory cells. So memory cells will produce the antibodies that are specific to that particular pathogen as fast as possible before you even realize you're sick. And this is the importance of suffering from a disease for the first time and then you get it. So this way, it also works with the immunity, the type of immunity that we take. For example, the first time you are vaccinated, you are being given that particular disease in your body, and its role is to help your body learn the structure of the pathogen that may come, and then what happens next? You produce memory cells that when the real one comes, for example, the COVID, when the real one comes, the body has already produced memory cells and they are rapid and massive. So the immune response will be so high. Immune response refers to the ability of this body to produce antibodies within the shortest time possible and fight the infection before it causes any symptoms and diseases. And this is how this graph is explained. So what about vaccinations? This is what I've just mentioned. What happens with the vaccination? A vaccine, we introduce either a mild or dead form of the pathogen that we know is affecting people into a person that causes the production of antibodies without the symptoms of the disease. For example, you see how people are producing astrophinaco and Pfizer for different people as a vaccine for COVID. What they are doing in this, they are injecting people with a dead strain of the COVID-19 virus, so that once it goes into your body, your lymphocytes will learn the structure, and when they learn the structure, they'll produce antibodies that are specific to that virus, and produce memory cells without even you knowing that you're sick. So there is no immune response. So it's the same thing. The level of body antibody in the blood. So when they vaccinate you for the first time, first initial vaccination with mild or dead pathogen, what happens? The immune response is still going to be like the previous one. Antibody is produced, but they will be very low and the number will be very small. So in this process, two things are happening. We are producing antibodies to fight against that which is already dead, and also produce memory cells that will stay there. If the real one comes, then you see that the moment the second thing comes, the real one, that is the infection with the real of virulent virus pathogen, you see that because of the memory cells that are available, immune response will be so high. So by the time you land on someone with COVID, your body will produce a lot. Your body will produce these cells, memory cells, will produce massive antibodies. And those antibodies will fight the virus before even you get the clinical symptoms. So what is, for starters, what we have to know? That reason why we are vaccinated, a vaccine contains either a dead pathogen or a mild pathogen, which you call an attenuated pathogen or virus. Attenuated pathogen is when a pathogen is changed from harmful to harmless. And then they inject it into the body. When they inject it into the body, the roles are two. It will help the body, first of all, to understand such kind of a pathogen exists and produces antibodies to fight the one that is in the body, and also produce memory cells. So memory cells will help in immune response. That is, when the real one, the virulent pathogen is coming without your notice, this time it's not by injection. So your body, because of the presence of the memory cells, there shall be rapid, massive production of antibodies which will fight that pathogen before you get to understand that this thing is there. So let's talk about blood clotting here, and everyone should be in position to describe a blood clotting. If you look at this structure here, this is what we have a niche, a group of wire-like structures which are surrounding this part. This is how our body looks when it has stopped bleeding. So this is called the blood clot. A blood clot starts when a body is introduced to. With blood clotting, we are basically looking at how, when you get a wound, bleeding starts. But after some time, bleeding stops. What exactly takes place? What exactly happens? So when a blood vessel is damaged, either by a cut or other means, blood clots for two reasons. One, reasons why we need blood to clot. One, to prevent leakage of blood, that is loss of blood. Number two, to prevent entry of pathogens. Why does blood clot like this? To prevent blood from oozing out, because once you lose your blood, you become anemic. Or to prevent entry of pathogens from outside into the body. So these are the two areas where why we need blood to form a clot. How clotting happens? So how clotting happens? There is a protein called fibrinogen, which is saturating in the blood. A protein fibrinogen is soluble in blood, which saturates in blood. At points of damage in blood vessels, this protein is converted to what we call fibrin. When it is in blood, it's not fibrin, it's called fibrinogen because it's soluble. But the moment you get a wound and blood gets exposed to air, fibrinogen in blood is going to be converted into fibrin fibers. The fibers you see, which are forming the clot around the wound, are what we call fibrin fibers. Fibrin fibers, which form a net-like a protein which catches the blood cells and forms a tangle or a clot. This you see in white is what you call the clot. It comes as a result of damage or exposure of blood to air, whereby fibrinogen, which is a soluble protein in blood, is going to be produced, and once it is produced, it will be converted actually into fibrin fibers. As you hear the word fibrin, it's a fiber like your hair fibers, which form a network of fibers around the wound and prevents these cells from escaping, then forms a network that prevents blood from oozing out. So anyone, if we ask you which protein is found in blood that forms fibers around the wound to prevent blood clotting, it's known as fibrinogen. Then when it's exposed to air, there are a lot of chemical changes that happen which result into fibrinogen being converted into fibrin fibers, which form a network of protein fibers that catches and tangles or prevents blood cells from oozing out and also prevents entry of this. So, blood clotting is the first step towards healing. But there are people who have a genetic disease known as hemophiliac. Hemophiliac is when an individual lacks the initiating factor to blood clotting. The initiating factor to blood clotting, once it is not there, you are suffering from a genetic condition known as hemophiliac. Hemophiliac. So what is happening here? Blood clot forming, the blood protein fibrin, which is white, acts like a net at the point of damage. It catches red blood cells and they form a clamp which blocks opening into the blood vessel, and that prevents the entry of pathogens and prevents the leakage or loss of blood. So if we ask you to answer that in multiple choice questions, that will happen. So what happens now? Sorry, just a minute. White blood cells move to the clot to kill bacteria or viruses entering through the point of damage. As you can see at this point where we have a damage or a wound where you've cut yourself using a knife, you hit yourself, you step on the nail, anything that happens to expose blood. So as the platelets accumulate in that area, you see that the number of platelets will become too much. That's why I told you their name is called thrombocytes, because they form an enzyme known as thrombokinase, which converts prothrombin to thrombin, which thrombin converts the soluble fibrinogen fiber to fibrin fibers, which fibers form a network of fibers around the wound. So as that is taking place, white blood cells are also running. So the white part you see are fibrin fibers. Then what we see next are the red blood cells. Then white blood cells will come at the entry here and they start fighting any invading, we call them the opportunistic infections that would take advantage of this wound. And this is why, as the wound is healing, it forms that thick white substance known as pus. Pus is a thick substance produced by these white blood by as a result of death. As so, pus is a fluid that is produced uh, as a result of the death of white blood cells and the viruses or whatever that has been fought here. So it's a combination of all the dead debris and all that stuff. That's why, as the wound is healing, there is pus which forms. That pus also joins to form the real part. That's the actually because of the clot forms the scar around the wound. So it's so unfortunate that some people remove that clot. When you remove that clot because you want a smooth skin, chances are high that you are making that body susceptible to be reinfected or to get reinfection. So we need to be very careful when we are dealing with those areas of ours when we get to do this. So when you look at this, we have this study was carried out by William Harvey, born in England in 1578. In 1628, Harvey published an anatomical study of the motion of the heart of the blood in animals, which explained how blood was pumped from the heart throughout the body, then returned to the heart and recirculated. The views this book expressed were very controversial and lost Harvey many patients, but it became the basis for all modern research on the heart and blood vessels. So credit goes to my brother William Harvey, who did this kind of work for some good time. So you can see the blood vessels here, how they move and all that stuff. So if we check this book, then you can see more. So I have what we call types of immunity, which we discussed there, or aspects of immunity. Among the immunity is the body's ability to defend itself against diseases. For example, we have the natural active immunity. So that is the immune system activation due to infection. So this is when you get an infection and then the body natural active immunity. You get the disease. After getting the disease, you suffer from it as the body B lymphocytes learn the structure of the pathogen causing that disease, then produce antibodies to fight that disease, and also produce memory cells which will help you to always fight it before you even know you have gotten it. So this is very good. It comes as a result of the presence of the memory cells. So that's what we call natural active immunity. When we look at artificial active immunity, this one comes with the vaccination, whereby the immune system activation is activated by vaccination. Why is the other one natural? Because you get that disease naturally, suffer, heal, produce memory cells. With artificial active immunity, that is the vaccination that we do. You inject me with a dead or halfway dead or an attenuated virus so that my body can learn from it and produce memory cells again. Then artificial passive immunity. This one is when you are sick, they inject you with antibodies used against potentially fatal and fast-acting disease, e.g. tetanus and antitoxins, but you never produce memory cells for it. It's artificial because it involves injection. Then we have what we call natural passive immunity. This is the easiest. This is the antibody. We get antibodies which are known to long lasting from our parents via breast milk, via the placenta, whatever, colostrum that prevents the virus from causing infections. So what I want everyone to pick, when you see the word natural, you get it by natural means. That means no need of infection, no need of injection, but it's either by infection, it's by infection. Then artificial, that one is by infection. So natural immunity is of two, natural active, natural passive. Natural active, suffer from the disease, get the disease fall sick. The body learns how to deal with the disease, gets the defenders, use them, and produce also memory cells, which will stay. That's why it's called natural active immunity. Then natural passive. It is natural because you get it from your parents, you get it from anything, and then with the natural passive, you get it from your parents naturally, via the placenta, via the colostrum, which is milk. Then when we come to artificial active, we are stimulating your body to produce memory cells by injecting it with that vaccine, with that vaccine, with that either the pathogen or. And with artificial passive, this is injecting you against a disease which is killing you. You get treated, but you don't need to. It will not produce memory cells for any more time, for any more way. So that is injection with artificial injection. Then, if you feel you like my lessons, you can subscribe and also do more of these videos. Thank you so much for now.